I think we're getting ready to start. Uh, it is 8:05. Thank you all for joining our second Biotech Connector speaker series in 2023. Welcome to our audience here and the folks online. I know we have quite a few that are joining online. I am Vladimir Popov. I'm the Chief Innovation Officer here at the Frederick National Lab for Cancer Research. Biotech Connector is the event, it's a collaborative event between the Frederick National Lab and Frederick County Chamber of Commerce. This event is an exclusive quarterly series that brings together scientists in a specific area of interest from the region and introduces scientists from the Frederick National Lab as well. Aside from sharing expertise and scientific curiosity and discoveries, this is also a great opportunity to network and to talk to other scientists and entrepreneurs. Little pitch about Frederick National Lab. It is a collaborative enterprise, and we're always interested in partnering and collaborating with other entities, whether they're small or big companies or academic institutions. We engage with other entities through collaborative efforts, through subcontracting, and so those subcontracting opportunities are available online. We also have a number of open positions, so if you're considering a career change or a new career, please check those online. I think I can change the slide here. We have a great event today, and the event is about the different imaging techniques and their applications in medical and cancer research. Let's cover some logistics first here. We'll move through the speakers, and we'll have time for a couple of questions after each speaker. When prompted, please stand up or come to the podium, or for those folks online, you can write your questions in the chat box. Now, it is my pleasure to introduce three amazing speakers that we have for today. In no order of importance, but in the order of presentations. First is Dr. Anna Alexandrova. Anna is the Advanced Workflow Specialist, confocal in Mid-Atlantic US at Leica Microsystems. Anna has over 10 years of hands-on lab work applying versatile microscopy, image analysis, molecular cloning, cell and embryo culture, CRISPR, mutagenesis, protein biochemistry, and other techniques to achieve high-quality scientific results across multiple research institutions in US and Canada. In her role at Leica Microsystems, Anna provides application and sales support to her customers in Maryland, Virginia, and DC. Second, we have Dr. Tommy Turbeville, one of ours, and he is the RAS Initiative Imaging Team Lead in Cancer Research Technology Program, also known as CRTP, here at the Frederick National Lab for Cancer Research. As a leader in imaging team, Tommy's current work focuses on characterizing KRAS in cell membranes using single molecule imaging techniques. His team is also actively involved in building assays to identify small molecules that disrupt KRAS activity in cells. Last but not least, we have Professor Emilia Encheva, and Emilia is the director of the Cardiac Optogenetics and Optical Imaging Laboratory at Department of Biomedical Engineering at George Washington University. Her research group combines biophotonics tools with human stem cell-derived cardiomyocytes technology and gene editing approaches to aid in advancement of personalized medicine. Now, it is my pleasure to welcome our first speaker, Dr. Anna Alexandrova.
All right, good morning, everybody. Can everybody hear me okay? Excellent. So it is my absolute pleasure to be here today. Thank you so much for the invitation. And we're going to talk about different methods of contrast in optical microscopy that can be used for cancer cell imaging or any other kind of biomedical research imaging one might choose to partake of. So, of course, we're all familiar with fluorescence. Fluorescence is a method to target one or more molecular targets in the specimen and then reveal the information about it in a precise, however, a little bit limited way. There is another aspect of the fluorescence process called fluorescence lifetime. We're going to talk a little bit about it today, that provides more functional information. Then there are nonlinear contrast techniques such as, for instance, second harmonic generation, third harmonic generation, that allow one to generate contrast in the sample that would be unlabeled based on its biophysical properties. Like, for instance, collagen molecules can be visualized this way. And then there is a very interesting form of contrast called chemical vibrational contrast that is based on the properties, vibrational properties of the chemical bonds found in the molecules. And we're going to talk a little bit about that today as well. And at Leica Microsystems, we believe that the synergy between all these different types of modalities can provide more information together than each one of them separately. So this is a very small example, one out of millions of published examples of the use of fluorescence imaging in cancer research. In this particular paper, the researchers visualized two populations of cells, fluorescently labeled, the LGR negative, LGR5 negative, and LGR5 positive cells. And they found that the LGR5 negative cells actually migrate. Yes, so LGR5 positive cells actually are the first to migrate during the metastasis process, and then the LGR5 positive cells join them later after the metastasis has been established. So just like this example, fluorescence provides very helpful spectral information. However, it can be further aided in the functional aspect of the information it provides by the temporal dimension of the fluorescence process. So we're all familiar with the Jablonski diagram, so the diagram that describes the transition of the sorry, I'm just going to show this. So that describes the transition of the fluorescent molecule from the ground state to the excited state after the absorption of the fluorophore, that then leads to the emission of the fluorescent signal of the photon with lower energy. So during the fluorescence imaging, we detect this photon. However, there is another aspect of this process that we can probe during the fluorescence lifetime, during the imaging, and that aspect is the fluorescence lifetime. So the time that the molecule spends in this excited state is as characteristic of the fluorescence molecule properties as its emission profile. And this time is commonly defined as the fluorescence lifetime. The fluorescence lifetime, interestingly, is very characteristic of the microenvironment that fluorescence molecule finds itself in, such as pH, temperature, ion concentration, or the presence of any binding partners in the area. And as such, there are instruments such as, for instance, like a Stellaris 8 Falcon system that allow one to measure the lifetime by virtue of having pulsed lasers with very precisely defined timing of emission, and the fast and sensitive detectors that again, very precisely measure the time at which each photon arrives at the detector. The time difference between those two events, when measured over millions and millions of iterations, can actually yield a measure of fluorescence lifetime that can be then used to characterize a specimen. This process is commonly referred to as time-correlated single photon counting. And as a result of the fluorescence lifetime imaging, one can generate a representative image whereby each pixel is not only encoded by its intensity information, but it also has the lifetime information associated with it. Like, for instance, this cross-section of the mouse embryo that clearly has multiple domains with different lifetimes present here. And as such, FLIM creates images with lifetime information. The common applications of fluorescence lifetime imaging are studies of molecular interaction, biosensing of different ions, DNA compaction, and other types of changes in the tissues, and species separation for the component analysis. And as such, FLIM is a great tool for functional imaging. So this is just one example of many of the use of FLIM for cancer biology. Researchers here researchers in this that generated this organoid. Unfortunately, the movie is not playing, but we'll just work with it as a still image. So basically, this is the cancer-derived organoid, and it incorporates the FLIM-FRET biosensor that looks at the interaction between ERK kinase and its downstream targets. And basically, the higher the degree of interaction, the more red the cell appears on this image, and you can see that there's a significant enrichment of ERK activity in some cells, whereas some cells show a decrease in such activity. So this way they were able to look at the heterogeneity of ERK activity in these organoid populations. In this study, the researchers actually looked at the property of the DNA dye that we're all familiar with called Hurst, that actually changes its lifetime based on the degree of the DNA compaction in the cells that it finds itself in. And they actually found that in the cells that display stem cell properties, DNA is a lot more compacted, and the Hurst in those cells displays longer lifetimes than in the surrounding cells. So it was a useful biomarker for them to detect the stem cells in organoids. Leica Microsystems provides a variety of tools for probing the fluorescence lifetime in cells or living organisms. And there's a suite of tools called TauSense tools that I won't go into detail as to their individual characteristics, but there is an application note published in Nature that describes these tools. I will just show you one use case example. So as you can see here, this is a movie of that highlights vesicular transport in cells. The membranes of these cells are labeled with the near IR dye that happens to be pH sensitive. And as you know, the endosomes, as they mature from early endosomes to late endosomes, the pH in those organoids changes significantly from 7 to 4. If you just look at the fluorescence image, you cannot really discern individual populations of this maturing endosomes. However, if you use a tool called TauSense, that can allow you to bin different lifetimes that are present in the image for different subpopulations of endosomes at different stage of maturity, you can actually very clearly discern individual subpopulations of those endosomes. And when all of these lifetime or average arrival time derived channels are put together, then you can really start seeing the interactions between endosomes at different degrees of maturity, and even fusion events between them that change the pH of this fused organoid. So that's quite interesting. And then in the last remaining few minutes, I would like to talk to you about the methods for vibrational contrast in that can be used for cancer or other biomedical research imaging. So vibrational contrast methods are largely based on the Raman effect. That is the biophysical property whereby if a laser light is shown on the biological specimen that is not strong enough or doesn't have the proper wavelength to actually excite fluorescence in this tissue, then the light largely passes through the sample. However, there is a small probability that the some portion of this light will actually excite the vibration in chemical bonds present in the sample. And because some of the energy was used up to generate this vibration, the there's a Stokes photon, the lower energy photon, that gets emitted in the process, and that can actually be detected. And on the electron level, the molecule basically moves from the ground state to a virtual state, which is lower energetically than the excited electronic state. And when the Stokes photon is released, then it returns back to the ground state. These spontaneous Raman events are very rare, so we had to engineer a way, or researchers had to engineer a way to make them happen with higher probability. And that was described as coherent Raman scattering, whereby in addition to the pump laser, you actually introduce a Stokes laser. And the difference in frequency between these two laser lines is engineered in such a way that it precisely matches the vibrational frequency that you expect from the predominant biochemical contributors to your specimen. And as such, so both of those lasers are, of course, introduced through the microscope objective. And as such, the populations, the vast majority of the molecular population in the sample engages in an event called vibrational resonance. And that can be measured, so this event can be measured either in the form of loss of pump beam and gain in Stokes beam, and that is referred to as stimulated Raman scattering, or there is a nonlinear photon absorption event that also takes place during this process that can be measured as coherent anti-Stokes Raman scattering. In both of those cases, basically the contrast is generated in otherwise unlabeled specimens that can be used for contrast. And this is one example of that. Basically, this is an organoid where lipids and proteins are unlabeled but visualized by means of SRS. And you can see here that you can very nicely see the cellular membranes and outlines of nuclei. This example shows that you can actually combine in a multimodal fashion the SRS and CARS imaging with second harmonic generation and general confocal contrast. This shows the unlabeled zebrafish eye, so you can actually use CARS and SRS in intact model organisms if you so choose. Then, as far as the applications for the technology, it is commonly used in cancer research, and we'll just very briefly talk about this example. It can be used in pharmacology, whereby if a drug has a certain Raman signature, then one can look at its redistribution and metabolism in the tissue. And then there is, of course, a use for general biomedical imaging, like, for instance, this cross-section of unlabeled mouse brain, where you can see the amyloid plaque as well as normal neurons. So as far as one particular use case in cancer research, this group in Germany generated these tricellular cancer spheroids that contain melanoma cells, keratinocytes, and fibroblasts. And they used CRS imaging to visualize lipids, saturated lipids, and then second harmonic to visualize collagen. And as you can see here, they were quite excited to find an unexpected cell phenotype inside the core of this organoid that actually was very enriched in lipids. And then they went on to biochemically characterize the vibrational frequency profile of different populations inside this cancer organoid. Furthermore, they were able to look at the redistribution of the chemotherapy drug after that was administered onto the cells, all using the coherent Raman scattering. And with that, thank you so much for your attention, and I'll take any questions if there's time. Yeah.
So, in the CRS,
Mhm.
so every big number gets a tumor, it's self-evident that the
So you can pseudo color it, right? So you get a grayscale channel, and then you can choose a color for each wave number if you so choose. Yes. Yes.
So I know this has been talk about preclinical work, but as you know, in clinic, many times lifetime spectroscopy and other tools are used for superior imaging over white light microscopy, white light imaging, and colonoscopy or bronchoscopy. Of these new technologies that you spoke about, are any of them amenable to clinical application?
This is an excellent question. I don't see the reason why they wouldn't be. However, some of them, like for instance, fluorescence lifetime imaging, has the same constraints as your common confocal imaging, right? Depth of penetration and such. So if you're looking at superior tissue layers, then yes. But if you're trying to go deeper, then perhaps with some multiphoton approaches that will again allow you to go a few millimeters deep. It's all a matter of biophysical constraints of light penetration in this case.
You could envision, say, in Mohs surgery, where you're looking at single cell depth layer changes, that it might be amenable and help guide the surgeon. That's what I was sort of wondering about.
Absolutely. So these methods are definitely fast. So they both CRS and lifetime are available at video rates. So it's just a matter of, I guess, engineering a microscope that can perform these modalities yet be able to access a patient or tissue. All right. Thank you all very much.
Oh. All right. My name is Tommy Turbyville, and I'm really happy to be here today to talk to you about some of the work that we do here within the RAS Initiative. So the RAS Initiative is about 70 scientists working on one particular problem in cancer, which is a mutation that happens in a gene called RAS. So RAS is commonly mutated in a number of different cancers. And I'm just highlighting a few here. Most commonly, KRAS, which is our main focus, is mutated in lung cancer, pancreatic cancer, and colorectal cancer. So both colorectal cancer and lung cancer are the most common cancers, or one of the most common cancers. And then pancreatic cancer is one of the most deadly cancers. So this is a pretty severe disease burden in the human population around the world. And in addition, there are other cancers that have different mutations. And this just shows the distribution of the mutations in different isoforms of RAS, but it's basically the same kind of protein that's mutated in cancer. So I'm going to give you a little bit of background of what RAS is so that you have a general understanding. So you can understand RAS as basically a little molecular switch, like a light switch. And this over here is what it looks like structurally around about. It's just a small protein that inserts into the plasma membrane of cells. So all cells have a plasma membrane around them that separates the outside of the cell from the inside of the cell. And it sits on the inside of the membrane. And what happens is when growth factors come from the outside of the cell and bind their receptors, it stimulates the activation of RAS. So it turns on the switch in the normal situation, in the normal tissue. And then what happens then is that RAS activates signaling cascades that are downstream. And that turns on things like cell growth and proliferation. So you can imagine what happens if this switch gets stuck in the on position, right? So right here, this is just demonstrating the fact that normally RAS switches between on and off, and it's totally dependent on receiving a signal from the outside of the cell to tell it to turn on. Okay, so normally it turns on, it signals downstream, and then there are things that turn it off. Okay, so that's a normal function. In the mutant function in cancer, what happens is it gets stuck in the on position. It can't be turned off, as indicated by the thinness of this arrow right here. So it's stuck in this on position, and as a consequence, it just keeps signaling downstream through these pathways. And so you just get constitutive activation of these pathways. You get cell growth, cell division as a consequence, and so you start to get a tumor. This here is an example of the way that cancer forms in a tissue, in pancreatic tissue. So one of the first mutations to happen in the lifetime of a person who has pancreatic cancer is for RAS to be mutated. And then there are a number of other mutations that will happen over years. So it takes years for cancer to develop. It's not an overnight thing, and it's not just one gene that causes cancer, but RAS in this case is one of the driver mutations of pancreatic cancer. So eventually the tumor starts to grow abnormally and metastasize, and that's when it becomes really dangerous. This is a statistic from the NCI SEER. It's a surveillance program that they have for monitoring cancer in the population. And this just shows you how deadly pancreatic cancer is. So this statistic right here has changed a little bit since I've started doing this work. It's gotten a little better. It used to be 5%. Now it's 12%. So, but that's still a pretty dismal statistic. So after 5 years, a pancreatic cancer population out of 100, there are only 12 people still alive. So it's a deadly disease. And we really need to find solutions for this kind of process, cancer. All right, so I told you already that RAS goes to the membrane of cells, and these are two images of pancreatic cancer cells in the lab. So we're able to take cancer cells and grow them on tissue culture, on glass, and then we're able to visualize RAS in the cell. So as you can see, the bright colored here, the bright colored protein is RAS in the plasma membrane. The tumor cells form a kind of cobblestone appearance. They're growing in 2D on glass, so they kind of grow together and they form these contacts with each other. So what you're seeing is a cobblestone of cells with RAS in the plasma membrane. So the plasma membrane is a very complex organelle. You can think of it as a 2D organelle. There are hundreds of lipids, different kinds of lipids in the plasma membrane. And then in addition to that, there are many, many proteins. You know, I told you about growth factor receptors, but there are many proteins that are in the plasma membrane. And these proteins and lipids cooperate to form a kind of architecture. It's sort of like a building with different, rooms and areas where you can go into and where you can't. And the model, the current thinking about this 2D architecture is that it's kind of like corrals with picket fences. So you can imagine each one of these little corrals here has proteins and lipids in it, and they're sort of bouncing around. And sometimes they hop out of these corrals into other corrals. And these corrals have different dimensions. So some are rather large, 400 to 300 nanometers, and some are smaller, all the way down to the size of proteins. So this right here is a simulation showing two RAS molecules that are about to interact in a lipid bilayer. So this gives you an idea of what that might look like. So we're able to image these things at the single molecule level using technologies such as what Anna was just describing. So we have a microscope upstairs. It's a turf microscope, which allows us to just visualize the proteins that are in the membrane of the cell. So this is just an example of a cell right here. A cell is microns thick. It's like 10 microns, 15 microns thick, but the membrane is only about 5 nanometers thick. So very, very small compared to the rest of the cell. The problem is when you're imaging a cell, you don't want all the background fluorescence from the rest of the cell, because then you won't be able to see single molecules. So using this turf system where you're bouncing a laser off of the cover slip so that it's at a certain angle, you're able to avoid getting light into your system, into your cell, but you can get a little wavefront of energy that goes about 100 nanometers into the cell. And that will excite fluorescence. And you can see those molecules very clearly. And we have these very bright dyes that are now commercially available. Actually, these that we use were invented at Genelia Farms, which is close by to here. These bright dyes are very photostable and bright, and so it allows us to image single molecules of RAS in the membrane of a cell. So this is a cell in which we're imaging RAS that's tagged with the fluorescent ligand that allows us to visualize individual molecules of RAS moving around. So we've taken advantage of this to be able to image RAS at the single molecule level and then generate maps of how RAS moves laterally in the membrane. And this gives us a lot of information. Let me just see if I can turn this. I don't have control over the, oh here, this is what I want. So within this region of interest, which is about 10 microns on a side, we're able to look at individual molecules of RAS, and then we're able to localize those molecules to very high precision, and then we're able to generate maps, 2D maps of how that protein is diffusing in the plasma membrane. And we're then able to generate a landscape of how that molecule is exploring the membrane. And this is just showing in 3D representation, the hills and valleys of RAS diffusion. So sometimes it's moving fast, as indicated by the yellow areas, and sometimes it's moving really slow, as indicated by the blue areas. And that gives us some indication of what RAS is doing in those different environments. When it's moving fast, it's just interacting with lipids. When it's moving slow, it's interacting with other proteins, probably signaling. And we've been able to show that with a number of studies. In addition, my colleague Rebecca here has been making synthetic bilayers. So we can take lipids and then we can assemble them into an artificial lipid bilayer and put that on a glass cover slide. And then using the same imaging technology, we're able to image single molecules in a supported lipid bilayer. And so this is just a cartoon of what that looks like, of RAS molecules interacting, forming little nanoclusters and interacting with the lipids underneath. And then we can measure the diffusion of those molecules and we can analyze that. This is just a plot that shows the characteristic diffusion behavior. So that bend that you see in that curve is an indication that it's more confined or it's interacting with more things in the membrane. It's not randomly diffusing. And we can see in this map on the far left or right from your point of view, it sort of shows you the diffusion map, that it's very heterogeneous. So this becomes important for a number of different reasons. We're able to characterize the biology of RAS, and as it diffuses, we can combine it with two-color imaging to look at some of the things that it's interacting with, other proteins and other lipids. And then we can also do drug studies. So this is just one example of many that we've done using this technology. It's not high throughput, but it gives us quite a bit of information. So this drug, BI-2852, is a compound that Boehringer Ingelheim, which is a pharmaceutical company, developed. It binds RAS directly. And when it binds RAS, it actually forces it into an artificial dimer. So we've taken this molecule and we've treated RAS proteins that are in a lipid bilayer, an artificial lipid bilayer, and then we've tracked those molecules. And when we track them, we see that they behave slightly differently. You can't tell by eye, easily, but you can quantitate those differences and generate a curve that looks like this. So this is just looking at the step length. That's how far the molecules move in a given time frame, okay? And what you see is that the step length shortens as you increase the concentration of this drug. So the drug is binding directly to RAS, forming these artificial dimers. As you increase the concentration, you get more and more of these dimers of RAS, and so it's slowing down its diffusion. It's It's shortening the steps that it can take in a given time frame. So this is just one example of what we can use this technology for. And this is just one example of the kind of thing that we do in the lab. We do a lot of different other experiments that allow us to screen for small molecules that are binding RAS in cells, and then image that in other different ways. So I think that's, in summary, what I'm here to talk about today. And if you have any questions, I'm happy to take them. I just want to acknowledge, though, that this involves a lot of people. So I just showed you a snapshot, but there's a lot of people in the imaging lab, both current and former members, who do a lot of work. The image analysis is a very big part of what we do. So quantifying these images takes time and takes specialized software and expertise. And then on top of that, we rely on a whole group of people here who make proteins and constructs, DNA constructs and things like that, that we use in cells or in our supported lipid bilayers. And so this is a very collaborative environment here. We work together. We also work externally with many people in pharmaceutical companies, biotech, academia across the country, actually around the world. So we're a very collaborative group, and I'm just one small little part of that. All right, thank you. Any questions? Yes, please.
So Tommy, really elegant science. I was just wondering, do you ever become concerned that the tagging of the RAS molecule will change its physiology?
Well, we address that. We do controls. So for instance, when we express the tagged protein in cells and then stimulate with growth factor, we can see downstream signaling. So it can recapitulate the signaling pathway. We have these MEFs, mouse embryonic fibroblasts, that only express one isoform of RAS at a time. And we've replaced the tagged version of the protein in those cells, and they're able to proliferate and grow. So those are the kinds of controls we do, just to make sure that it's able to signal and support the growth of the cell.
But if you consider directly tagging the molecule in a different way, and then seeing they'll deregulate the physiology?
Well, we've definitely been able to directly label RAS with fluorophores, for the synthetic experiments, but in cells that's a little trickier. Yeah. Yeah.
I'm not like a very high professional in this area, so I want to apologize if my question is too.
Please.
But my question is that I understand you tag the molecules, but how you separate ones that are mutated from non-mutated ones? As I understand, in cancer cells, it can have some proteins that have this mutation, some proteins they don't. So how you make this benchmark that you are sure that you work only with ones that have the mutation and other ones are not included?
Yeah, that's a great question. So we use genetic engineering to make sure that the protein that we're looking at is the mutant or the wild type, the unmutated version. So we're able to generate a DNA construct that has RAS, the protein, the gene that codes for the protein, and then we tag it with a fluorescent protein, for instance. And then we express that uniquely in the cell. And then we're able to see just that fluorescently tagged protein and not the other ones. But we're always dealing with fluorescent technologies like this with a lot of dark matter. Okay, just like in astronomy, people don't see everything. So fluorescence microscopy is limited because it can only look at 2 or 3 or 4 things at once. And then you're missing all of this information. There are hundreds of different lipids and many, many thousands of proteins. And, so you're actually missing a lot of information, which is why that Raman spectroscopy can be very powerful, right? Because that allows you to see more the context of what's happening in the cell and discriminate the different constituents of the cell a little bit.
But
yeah.
Then you test certain drugs on those proteins that they target those proteins, but how you make it highly specific? Because it can be the same mechanism that they target healthy RAS, mutated RAS. So I'm just curious.
Yeah, no, those are excellent questions. Those are the exact kind of questions that people who are developing drugs here and elsewhere for RAS have to answer. So, how specific is the molecule? Is it only binding the mutant version versus the wild type? There are a number of pharmacological things that you can do to discriminate. You can use different kinds of cell lines. You can use cell lines that are engineered to express only one isoform, one form of the protein, and make sure that it has specificity and that it's uniquely active against what you're hoping to achieve, that selectivity against. But most drugs have some dirtiness. They're not quite as specific as people imagine or hope, because the human body is so complex. But there are very good targeted therapies. So I don't want to downplay the effort that so many people are on to try to develop targeted therapies. There have been successes, including molecules that bind directly to RAS and inhibit it.
So there are ways to find this kind of special binding sites that only bind to genetically mutated RAS than healthy ones.
Yeah. So the additional thing to say is there are lots of biochemical, structural, and biophysical techniques that we can use that take the protein out of the cell, purify it, and do experiments to make sure we actually know exactly which atoms are interacting with the drug, and can map that out very, very specifically, and then compare that to an unmutated version of the protein, for instance.
My last question, for example, if this cancer cell has this mutated version, are all of them mutated, or there are still like a percentage? For example, this is a cancer cell that has this mutated KRAS, right?
Yeah.
And does it have all of them mutated, or it has some mutated and some not mutated?
That's a great question, and tumor biology question. So in patients who have pancreatic cancer, 95% of the cancer patients have mutations in RAS because it's one of the driver mutations. It's one of the things that initiates and starts the cell along the path to becoming a cancer cell.
There are still 5% that are still cancerous, but they don't have it, right?
They have some other mutations that are not RAS specific. Yeah, but generally speaking, there are different kinds of mutations. There are the driver mutations, and then there are mutations that are sort of passengers. They just come along because the tumor cell evolves and is dividing quickly, and there are lots of mutations accumulating, but they're not really responsible for driving the cancer. But there are some mutations that are really driving the cancer, and those tend to be in all of the cells. They're in the cancer cells. But it's a complex question, and it's a good one. Yeah.
I had a related question. The data that you showed, was that from wild type RAS? And if so, do you see differences if you express mutant RAS pathway?
Yeah, we've done those experiments. So the mutant RAS actually slows down because it's interacting more with effector molecules. So we can measure that effect. We can measure that mutant RAS interacts more with effectors downstream, and so slows down. Yeah.
Have you ever tried to look at mutant cultures?
No, be interested in trying that. It's a little bit difficult to do that with turf microscopy, but yeah. Yeah. Any other questions? I want to leave time for the next speaker. Yeah. All right. Thank you, everyone.
Hi, it's a pleasure to be here. I'm a cardiac researcher and will try to make links to cancer as much as I can. So at the heart of understanding biology, you can think of functional genomics, where we would like to know the role of different genes. Let me laser pointer. If we perturb a gene, how does it affect the phenotype? That's at the crux of our understanding of biology. Most of our knowledge over the years has come from transgenic mice, where a gene is knocked down, knocked in, and then we studied the phenotype, what happens. In the recent decade or so, there is a lot of push to move to human models, experimental models, and understanding. And last year, FDA announced that it will no longer require animal tests before going to human drug trials. It sounds very profound change. The law that required animal testing in preclinical trials was passed in 1938. And this is a recent announcement. So what are the alternatives if we develop some new therapeutics? What are the options if we exclude animal testings? Well, the options are to go in silico, to have sufficient knowledge to predict the action of a therapy or a drug, or to use in vitro human-based models. For cancer, you can extract a tumor and study it. For cardiac applications, we don't have that luxury, so it's very hard to come across experimental cardiac models. So here comes stem cell technology, particularly induced pluripotent stem cells, which came around 2007. And this is one vision how the current pipeline for drug testing can be possibly replaced by human-based testing, patient-derived iPS stem cell-derived cardiomyocytes in this case, that possibly could better capture different aspects of the population and reaction to the therapeutics, such as ethnicity, gender, cardiovascular history, and so on. Keep in mind that any therapeutics that is developed for cancer, for nail fungus, whatever, is required to undergo cardiotoxicity testing, any drug that will go to market. So there is a great need to have the right experimental tools to help reduce cost and speed up drug to market. And here comes the technology that we have been interested in developing for the last several years. So cardiac cells exist in a continuum, in very tightly coupled, very structured environment in the heart. This is an image from my lab in a mouse heart, labeled with a voltage sensitive dye that happens to label the membrane and the T-tubules. So the current cardiotoxicity testing is done with patch clamp technology, which also underwent a lot of nice developments. Planar patch clamp, high throughputness has been implemented there. But those are single cells that are dying single cells. The moment you patch a cell, it's on the way to die. So we believe that optical technology can give us tools to study cells in context while they're connected to other cells and more physiologically relevant. About 15 years ago, my lab got interested in translating optogenetics, which emerged 2005 in neuroscience. We wanted to bring it to cardiac applications. And so optogenetics are these light-sensitive proteins that we borrow from algae, from bacteria, which there are a range of them, but particularly interesting are the ones that have millisecond response time. So you can use them to actuate things, to start biological process on the millisecond time scale, or to image biological process with the same precision. The ability to encode them, we can make them cell-specific. So we can put them, let's say, in macrophages and study if I activate the macrophages only, what are the responses? Or the autonomic nervous system can be specifically made to be light sensitive and very precise control. In the clinic, one can envision this optogenetic-based tools to be used for optical pacemaking, optical cardioversion, defibrillation. But we have to keep in mind that it requires the delivery of the construct, so it requires technically gene therapy and some access of light to the location. There are clinical trials for vision restoration with optogenetic tools that have been deemed safe and even shown some efficacy. But the near-term translation of this technology we see in parallelizing means for drug testing, preclinical means for drug testing. The high throughput systems that we were interested in developing, we started with microscopy, with modifying an inverted microscope to be able to stimulate by light. We use blue light to stimulate the cells that are genetically modified to respond to it. We use green light to image calcium in these cells, red light using near-infrared dyes to image voltage. So we map multidimensionally things. In our first implementation, we went sequentially, well by well, in high throughput, in this 96 well format. And these signals recorded from iPS-derived cardiomyocytes, action potentials, voltage, and calcium. Disruption in these signals would be indicative of cardiotoxicity, and we have metrics for that. More recently, like early this year, we published 2 papers where we built standalone systems for studying the action of drugs in these cardiomyocytes. In the top here, we are using oblique illumination, which gives us means for label-free imaging of waves in the myocytes of electrical activity, mechanical waves actually. And we have validated that those mechanical waves, these activation maps, are similar to the electrical waves that we can image simultaneously with voltage-based dyes. We can build these systems very cheaply using computer vision cameras, machine vision cameras, CMOS cameras that are $300. Most of the camera technology in our field, because this is fast demanding imaging under low light conditions, up to 100K. So from 100K to $300, it's a big change. And most recently, we are building standalone systems, again, very cheaply, that can image whole plates to stimulate by light, to record voltage and calcium, very, very cheaply built. Other high throughput technologies that we couple to this, we develop means to stimulate without genetically modifying the cells by magnetically pulling spheroids that we built that have the optogenetic tools, and then we can use those as point stimulators. We built in collaboration microfluidic systems that can be coupled with standard plates. Here is a cover that we can, it's a microfluidics-based cover to perfuse standard 96 well plates while we are imaging. And this is more recent work where we do oxygen imaging of pericellular oxygen, which might be of interest to cancer research as well, where hypoxia is central. In this system, we deploy it in standard 96 well plates, glass bottom, where from the bottom we can do high-level imaging, and this half circle shapes actually oxygen sensors. The cells grow on top of the oxygen sensors, and in the other part, we can monitor for days in the incubator and detect the dynamics of pericellular oxygen as the cells are thriving, and then couple that with our all optical imaging of electrical and calcium activity. We have developed some pipelines how to modify the cells, grow them in this high throughput plates, apply our tools, then lyse them, and in these very small volumes, do qPCR and protein analysis in order to be able to correlate what I told you, gene function, gene expression to some functional parameters such as conduction velocity, which normally is difficult to do. So we do computational mapping of these relationships. One of my students invested in trying to develop interference or activation CRISPR in this system, couple it with our optical tools. So perturb one gene at a time in order to link to the phenotypes that we measure. And down the road, we would like to explore gene regulation networks and develop more understanding. We used a CRISPR-I system. So this is deactivated Cas9 with a Crab effector, which we put in already differentiated cardiomyocytes. And then as a proof of principle, we targeted some key cardiac ion channels. For cardiotoxicity testing, this hERG channel is essential. It's a very sticky protein where all the drugs seem to like to block it, and is essential for repolarization and for preventing arrhythmias. So it's a key in cardiotoxicity testing. It's a multi-day process to develop these assays. So Julie optimized the system, and we add optogenetic tools and the CRISPR-I, and we run this all optical electrophysiology, after which we can do fluorescence microscopy and molecular assays and correlate things. Here is an example of inducibly expressing dCas9, here with an mCherry reporter, and delivering guide RNAs to affect this key protein hERG. And then we see that much reduction in hERG at the mRNA level. It's not great, but these are already differentiated myocytes, and we are applying CRISPR-based tools in them, which is not normally done. Here, it's a busy slide, but I will draw your attention to a functional parameter. This is action potential duration, which prolongation in it is a measure of cardiotoxicity in some way. So we see that as we reduce the expression of this hERG gene, action potential duration is increasing in general. It's not a clean relationship. If we integrate our multifunctional assay parameters, we measure about 24 of them in voltage and calcium, we can build classifiers that are more sophisticated means of separating a population with reduced hERG and non-reduced hERG. So this could be useful for speedy preclinical testing. One more example where these colorful maps, activation maps of waves, and the more colors you see, the slower the waves go. The top is controls, and the bottom is with CRISPR-I on this gap junctional gene that connects cardiomyocytes. And it's frequency dependent slowing of the propagation of the waves. So the faster we pace, the slower the waves and more pro-arrhythmic this is. Again, we can correlate the gene expression or protein levels to some sophisticated functional parameter like the conduction of the waves. And a last slide, there is interest to go away from monolayers of cultures. I'm sure it's the same in the cancer research, the same in the cardiac research. So as a proof of principle, we actually used electrospun scaffolds within the 96 well plates, in some cases that are randomly oriented, and in some cases where we enforce orientation of the growth of our cells, and try to apply this for drug testing. Shown here are several drugs. Nifedipine is a drug that is safe and shortens action potential duration. Dufetalide is a known cardiotoxic drug. We use it here at very low concentration. So that's the red bar, which prolongs action potential duration reliably, pro-arrhythmic. And then vanoxarine is this interesting compound that has been around since the 1980s as a, I think, dopamine reuptake inhibitor. But 5 years ago, someone wanted to redevelop it as an antiarrhythmic drug, and it failed miserably in phase 3 clinical trials. It was very pro-arrhythmic. All those testings in animals did not capture that. We very clearly capture it because it's prolonging more than our positive control. I will stop here in interest of time. Julie and Yuli were the two that I presented the most data from, but this is my group, and thank you for your attention. Yes.
So there's one question to the audience.
Hi.
Gemma. How is the technique of all optical electrophysiology derived from the neurons? And what is the success rate on neurons?
Yeah, so almost simultaneously as our efforts, practically simultaneously, Adam Cohen at Harvard developed similar technology. There is a successful company based off that, Q-State Biosciences, which works exclusively in the neuroscience domain for drug testing. Judging by the success of the company, I would say it has made an impact. There is the so-called SIPA initiative, which is testing arrhythmia in a dish, led by the FDA and some international organizations, trying to push things for pharma to go more to human-based testing, to in silico testing. It's a very inert system. People don't want to change their ways. So it's a slow process because there are whole divisions that have been trained in, let's say, dog studies or isolated myocytes. So it's a very slow moving field, but makes an impact.
Has there been licensing interest in this? I mean, I was at NHLBI for a while. Opportunity for business of this full business support.
Yeah, we have filed several patents through GW. It's very active tech transfer office. And we have gotten interest from companies, mostly for know-how extraction. No one has licensed it yet, but there's definitely interest, particularly with the latest decision by the FDA that you can surpass animal testing. So, yeah.
I was struck by your last image where you showed that you could recapitulate the toxicity in vitro. Extensively, FDA wrote the stance. Kind of a question I was wondering about the clinic, in the cancer clinic. We know that it ducts roots and causes dose-limited cardiac toxicity, and the drug still works, but we have to limit the use because of that toxicity, as you well know. So I was curious, have you thought about intervening to find preventive measures where you have a known cardiac toxic agent to see if you could intervene and prevent the toxicity to screen for potential compounds that you could then return to the clinic?
Yeah, we haven't quite done that, but we have been interested in the intersection with oncology and have done testing on HDAC inhibitors and trying to understand which ones would be more cardiotoxic than others. Not surprisingly, the pan-HDAC inhibitors are more cardiotoxic. In general, the toxicity is very low in those molecules, but we have been interested in various classes of molecules, but we haven't quite done how to learn how to actually counter it. It's an interesting application.
So when you're activating the optogenetic tool, are you interested in polarizing the cell by only activating the your optogenetic tools in one part of the cell, or are you sort of looking globally in the monolayer?
In this system, we're interested in reliably pacing by light because it's effective and quick, and it's equivalent to electrical but contactless. We have another project which we target organelles and is more dimerization based optogenetics, not the millisecond based. It's slower. So we are targeting lysosomes, mitochondria, ER, SR, and trying to engage compartments to come closer together because that has implications for calcium release events in cardiomyocytes. So it's a separate project where we are interested subcellularly, but not in this. In this is brute force pacing by light. Yeah. Okay. Thank you.
Right. So let's thank our amazing speakers again for delivering such high-level presentations. Thank you. I just want to make a few notes. So I would like to thank our sponsors, Mike B. Moore and Venic IP, for such an amazing breakfast. Yeah, thank you so much, and the Frederick County Chamber of Commerce for supporting this event. So the recording of the event will be available soon on our frederick.cancer.gov website, and the next Biotech Connector event is coming in August, so it will be in a protein production theme. So please stay tuned. Thank you so much for coming, and for those who connected online. Thank you.